Hi. So in previous session I have initiated the innate immunity, and I basically gave you a very overview or introduction why innate immunity is needed, okay. And I have also talked how the if the pathogen come in contact with the host, then how this will be defended, okay. There are several layers of uh, protection, okay, like anatomical barrier. Then if it breaches, then there will be another set of immune cells, mainly innate immune cells, they will come and remove. And if it is further not removed, then there will be a activation of those immune cell, and they basically sense the pathogen associated molecular pattern, okay. And then that subsequently eventually these microbes will be transported to the nearest lymph node in order to develop more specific immune response, which is mainly mediated by B and T lymphocytes, okay. So now let us move further about the innate immunity, more precisely or more deeply in the innate immunity, okay. So basically innate immunity is consists of various component, okay. Components are like here you can see there is a physical, mechanical or anatomical barrier. So I will talk in great length about this physical, mechanical or anatomical barriers, okay. Basically it is consist of our lining, the lining in the body. If you know that the skin is the largest external organ in our body, so over there is a variety of mechanisms are there which protect us from this microbial invasion, okay. There is a biochemical or chemical barrier. So it's a not just a kind of barrier, it's not like a wall of house, okay. The mechanical barrier is not only the mechanical barrier, this is live, okay. They produce some or other kind of chemicals, okay, this physical barrier, okay, and they defend against the entry of microbial pathogen, okay. So basically the molecule which is secreted by this barrier, we call it as a antimicrobial peptide or protein, or sometimes we also call it as a antimicrobial enzyme. There are some enzymes are also secreted. So these are basically here you can see MBP, it is a mannose binding protein, and this is basically a lectin family protein. I do not know, you may probably studied that lectins are all those protein which is binding with the sugar molecule, okay. So those proteins we call it as a lectin. So mannose binding protein, the name suggests that it is basically binding with the mannose which is present in the microbial pathogen. There is a CRP, C-reactive protein. This C-reactive protein is basically also produced in order to defend the microbial pathogen. There is a APP, this is nothing, it is a acute phase protein which is mainly synthesized by liver, and these proteins are also defending against microbial invasion. Similarly, there is a serum amyloid A, alpha 1 antitrypsin, human beta defensin, and there is an antimicrobial enzyme which is produced by a variety of cells, okay, which basically breaks down the bacterial cell wall. Probably you know that lysozyme act on peptidoglycan, and this enzyme basically breaks down the peptidoglycan, and then they eventually kill the microbial pathogen, okay. So these are the various biochemical and chemical barriers. Another, the most important biochemical barrier is complements. So, complements are the set of protein which basically synthesized in the liver, and this complement basically do three things which I have told you in previous session also. I will quickly tell that. They basically induce inflammation, okay. They also opsonize the target microbe so that it will be readily phagocytosed by phagocytic cells, okay. They will also make a kind of a complex which we call it as a membrane attack complex. So, this membrane attack complex basically makes a hole in the microbial pathogen, so that eventually this microbe will die, okay. So, complements we will discuss in more detail in subsequent session in innate immunity. There are some microRNA also against mainly RNA or DNA viruses, mainly RNA viruses. So, there are some microRNA. So, microRNA is nothing, it is a non-coding small RNA, okay. This non-coding small RNA plays a very important role in gene regulation, okay, expression of gene regulation, okay. They basically, in general, they bind to the 3' UTR of the transcript, and they regulate the expression of or further translation of this transcript, okay. So, some microRNA basically they either bind to the genome of virus, mainly RNA viruses, and they basically reduces the replication of viruses, okay. This is one way. Another way is that when there is a virus infection, this microRNA can bind to the negative regulator of innate immune signaling pathway. So, when it will bind to the negative regulator of innate immune signaling pathway, then what will happen? It will activate or it will induce more antiviral expression of gene. So, in this scenario, the microbial pathogen, or here I am talking RNA viruses, will their replication will be reduced, okay. So, these are some of biochemical or chemical barrier. Another barrier which plays a very important role is microbial or microbiological barriers, okay. So, if you remember my previous session, there are two kinds of interaction which is present in with the microbe, that is commensalism and another is mutualism. So, in commensalism and mutualism, the bacteria or the microbial pathogen or microbes, not pathogen, I will not say pathogen, they are microbes, okay. So, they interact with the host and in some or other way they give a benefit, okay, to the host. So, microbial barrier is playing a reasonably very important role in defense. So, they grow in huge number and they do not allow to grow the pathogenic microbes, okay. So, their outnumber is also important. In addition, they produce some substance which basically inhibit the growth of pathogenic microbial pathogenic microbes, okay. So, in that way this microbial barriers play an extremely important role in defense. Another is the cellular component. So, this cellular component is basically when I say innate immune cells, so basically all cells come in innate immune cell, that includes immune cell as well as non-immune cells, okay. Particularly NK cells are, so these are professional innate immune cells like macrophages, NK cells, dendritic cells, Now you know there are so many kinds of macrophages present in different organs. There is a two major kind of dendritic cells, although there is a three, but here I am telling two because they are playing important role in innate immunity. One is that conventional dendritic cells and another is plasmacytoid dendritic cells. FDCs are playing important role in adaptive immunity. Okay. And these cells basically express germline conserved or germline encoded receptor, which we call it as a pattern recognition receptor. Okay. And this pattern recognition receptor basically senses the products of metabolic pathway of this microbe, and then it will induce some kind of signaling, and the result of this signaling will be production of inflammatory cytokine, type 1 or type 3 interferons, and so on. So, okay. We will discuss this in a great detail. Okay. This pattern recognition receptor, there are several families of pattern recognition receptor. For example, toll-like receptor, there is a Rig-like receptor, there is a nod-like receptor. So, we will discuss all this family of pattern recognition receptor one by one in subsequent session. Okay. So, now let us go back to the physical and mechanical or anatomical barrier. So, here you can see how this mechanical barrier is playing very important role in defense. So, this mechanical barrier in skin is basically constituted with this epithelial cells, okay, joined by very tight junctions. Okay. They are very tightly packed. Okay. So, microbe cannot enter, it's a quite rigid, it's like that the wall is very strong, the wall of house is very strong, nothing can just go, even water cannot go if you make a proper wall. Okay. It's something like that, it's a wall or guard in our body. Okay. Besides this, in skin there is a longitudinal flow of air or fluid, okay, that prevents the entry of pathogen. In gut there will be a longitudinal flow of air and fluid. We will discuss about the mucus. So, in our gut there is a continuous flow of mucus, okay, that prevents the attachment of microbe or entry of the microbe. In lungs also there is a movement of mucus by cilia. So, cilia is keep on moving, they are keep on doing this job and make the mucus flow. That is another mechanical barrier. In eyes, nose and oral cavity, in eyes there is a tear. Okay. This tear is keep on flowing and keeping a kind of layer over our eyes. Okay. And this tear basically consists of lysozyme, if you maybe you have studied. Okay. In nasal cavity, nose, there will be a cilia. Okay. There will this is keep on moving. Okay. So, this is providing a kind of mechanical barrier. There is a chemical barrier in these places. In skin, there are some fatty acids produced by microbe as well as the cells, okay, and basically this does not allow the growth of other microbial pathogen. There is a production of some antimicrobial peptide like beta defensin, okay, and there is a one more protein which is produced by these cells is cathelicidin, okay, and this is these molecules are present in some kind of structure which we call it as a lamellar bodies, Okay. So, this is keep on throwing. Okay. I will talk more about the defensin and this cathelicidine, how it works in maybe subsequent session. Okay. In gut, we have you all are aware that in gut there is a low pH, there is a production of hydrochloric acid which inhibit the growth of any microbial organism, okay, any microbes including pathogen. Okay. There is an enzyme secreted in the gastric juice, which you know there is a variety of enzyme, pepsin, trypsin, okay, and there are some more enzymes. Okay. So, these things basically defend against this microbe, okay, microbe or microbial pathogen. In addition, there is some antimicrobial peptide like alpha defensin, which we call it as a cryptidins, regal, which we call it as a lacticidine, cathelicidine. So, all these antimicrobial peptides are also produced in the gut. There is some specialized cell you will see in subsequent session, we call it as a Paneth cell. Okay. So, these cells will basically produce these molecule and defend against these microbial pathogen. Okay. In lungs, there is a movement of mucus by cilia. In gut also there is a continuous production of mucus. There is some specialized cell which is present in the gut. We call it as a goblet cell. I will discuss in more detail. Okay. In lungs, there is also production of mucus, which is the movement is mediated by ciliary movement. Okay. There is some surfactant. There is a pulmonary surfactant, which basically, so surfactant is which reduces the surface tension. Okay. Like if you so you know the maybe the concept behind cleaning of cloth. Okay. We use detergent. Okay. So basically this will solubilize the fat. Okay. And this has a both polar and non-polar group, and they are basically acting like a surfactant. Okay. They remove this dirt. Okay. So something like that, it is not precisely that, but something like that. So pulmonary surfactant, there are some molecules. Okay. This also produce alpha defensin, and again cathelicidine is also present. Okay. In eyes and nose and oral cavities, there are as I told you, there are some enzyme in tear, okay, like lysozyme. In saliva also it is present, and that defend against this microbes, irrespective of pathogenic or non-pathogenic. Okay. Histatines are also present. These are proteins or antimicrobial peptide, and there is a beta defensin. So all these molecules are doing, they are continuously doing their job. That is why we do not fall sick. Okay. We rarely fall sick, right? Not every moment we are sick, right? Because these mechanical and chemical barrier are keep on working. In addition, there is a microbiological barrier which has a normal microbiota. Probably you might be noticed when you take antibiotic, at that time this normal microbiota are destroyed, some of them, not all, yeah, but majority, and that gives some problem, stomach upset and all those things. So the doctor prescribe, or now there is a lot of advertisement about the probiotics. So probiotic is, it's nothing, just providing those microbes, the spores of those microbe, in order to reconstitute or remake this destroyed normal microbiota. Okay. So, you can understand the importance of this microbiological barrier. Okay. Now, I will talk about the skin in more detail, is physical component of defense. So, here you can see that there is a multiple layer of tightly packed cell, which basically does not allow the entry of pathogen. Okay. And this skin is keep on shedding off. Okay. It is dead skin cells are keep on falling off, and whatever, if some microbe will attach, then that will be also thrown out. Okay. So, in that way this this is giving a protection, and this is mainly in epidermis. You remember that there is a one specialized phagocytic cells are there in skin, epidermal layer, okay, which we call it as a Langerhans cell. So, that there is a presence of dendritic cells in the epidermal layer, which phagocytose this pathogen, and these cells basically extended out among other cells in epidermis, forming a network of intercept invaders. Okay. This basically intercept the invaders. Okay. There is a dermis layer, there is a, you know, the structure of skin, probably you may remember, if not please take a look. It's a simple structure. There is a dermis layer which contain a protein fiber called collagen. Okay. And this basically, this dermis is giving a skin strength and permeability to resist the abrasion that could be introduced by the microbe, okay, or microbial pathogen. There is a biochemical component of defense in skin, and basically it is mediated by perspiration, okay, basically secreted by sweat gland. We have a glands, and that produce some salt which will inhibit the growth of microbes by drawing water. You know that concept of osmosis. Okay. So, if there will be a more salt, then this will draw the water from microbial cells. In that way, this will also initiate some kind of killing or defending against the microbial pathogen or microbes. There is some antimicrobial peptide, as I have told you in previous slide. There is a defensin, cathelicidin. I will tell the mechanism in later session. Okay. There is a antimicrobial enzymes like lysozyme, which destroy the bacterial cell wall, that is peptidoglycan. There is a production of sebum, okay, secreted by sebaceous or oil gland, okay, helps in keeping skin permeable and less likely to break or tear. This also maintain low pH in skin, so that there will be inhibition of these microbes. Okay. Here I will show you the skin is one of largest external organ, okay, and they have several layers. Okay. Here you can see that there is a stratum spinosum, stratum granulosum, and stratum lucidum, stratum corneum. So, this stratum corneum is keep on the dead, it is mainly dead cells. Okay. Stratum lucidum is basically below this corneum, and stratum granulosum is in between lucidium and spinosum, as you can see in this picture. And here if you notice, there is a yellow kind of laminar bodies, and these laminar bodies basically contain the beta defensin and cathelicidin. Okay. And this there will be a growth of this stratum spinosum, and that is pushing out. Okay. So, towards terminal there is a dead cells which is giving a kind of mechanical strength, and in addition this will also produce some kind of lipid layer, which is a watertight lipid layer, and in that way the microbial pathogen cannot enter. Okay. So, I will stop here for this session. In next session I will discuss about the gut, okay, what are the arrangements are there in the gut as well as in respiratory tract, and I will take it further. Thank you.